think we're gonna go ahead and get started with the afternoon session. We get people to come in and take their seats. You want to say something, Nikki?
Hi, everyone. Thank you so much. If everybody could, I think most everybody is back from lunch or trickling in. We're going to get started with our third session of the day, which is going to be presided over by Garth Ehrlich. Thank you, Garth. We are going to allocate time to questions. We appreciate everyone's patience. We're a little bit behind, but that probably only means like 10 minutes in the grand scheme. And what we can always do is, depending on how things go, we have 30 minutes at the end allocated to a panel discussion and questions. We can always shorten that, or we could go 10 minutes longer into 5:10 and go have a glass of wine a little bit on a delayed schedule. And for everyone online, we're sorry we can't give you a glass of wine. And I'm going to turn it over to Garth now.
Thanks, Nikki. Gives me just great pleasure to introduce our keynote speaker, Dr. Wenyuan Shi. Wenyuan is the director of the Forsyth Institute in Boston, which has recently formed a major collaboration with the ADA. I've known Wenyuan for a very long time, back when he used to be the chair of oral biology at UCLA, and his career has been distinguished by numerous major advances, both in technology and in development of that technology into commercial products, as well as being a wonderful basic scientist and colleague. And so with no further ado, Wenyuan, please.
Well, such an honor to be here. And Gus and I think we met back in 2006 or something. And it took 15 seconds. We instantly become a best friend because we have all the same curiosity and we have the same fearless attitude. And we were there to work on everything interested us. Now, through Gus, I got an opportunity to know Nikki and Brian, and I noticed, people always, the similar people gets together. And I just truly want to say, I can see the fearless, amazing leadership you're having. I really saluting what you're doing and what this is a very significant, meaningful, impactful, and timely. And so ADA Forsyth is very proud to be part of it. And I'm really hoping, through this interaction, I will have the opportunity and to work with you all. Now, I wouldn't be characterized myself as an Alzheimer disease expert, but given, it's a fearless nature and given life is full of surprise. So I'm there to taking on this and to have some communication with you all and see whether we can develop some collaboration. And life is a box of chocolate anyway. My child dream is to become a fortune teller. And that's how I actually decided to take a PhD in behavior genetics. And I thought that was a scientific fortune telling. And then I had the opportunity to work for Julius Adler, and some of your microbiologists know, and he studied the E. coli chemotaxis and become the founding father of the signal transduction. And to be able to study under Julius, the one thing, it was truly amazed me, at that time as a young biologist, and E. coli using 20% of its genome encoding intelligence. They got all that two-component system, the motility system, and doing all that. And through that study, I understand how smart bacteria is. And if we're going to be thinking like Lambda is one of the smartest molecular biologists, E. coli is 100 times better. And now we're looking at the microbiome, which is a social aspect of it. And the oral microbiome I'm going to tell you about. We got a 10 trillion bacteria, 700 different species, forming this most complex community and have incredible politics going on. And we can learn so much, through that. And that's what I, become a microbiologist. And I have to say, you know, even in my best crystal ball, I never thought I have anything to do with oral microbiology. And the only reason is when I was a professor at the medical school in the micro department, and as a replacement teacher, one day I was sent to teaching dental student oral microbiology. And that's where I said, aha, this is a jungle with no lion, monkey can become the king. So that's how my lab is, getting involved in study oral microbiology, even though initially triggered by the curiosity. But what we learned in the last 30 years is that this is just a truly amazing area. And for that reason, I was getting involved in the technology development and back at UCLA, and we funded multiple companies. So one of them, I managed to raise $250 million from the Delta Dental Insurance Company, which is now is a publicly traded company. So I become one of the few people to bordering between the science and the technology. And that's how 7 years ago I get a call, and a headhunter said, okay. So I get a call and said, and we're representing the Forsyth Institute. We're looking for a new CEO. Will you be interested in the job? My first reaction is Los Angeles time is great. I'm not interested in the job. But I say, you call me at the right time. My daughter is going to Wellesley. I'm going to track her down. But my real reason is, Forsyth is a truly amazing place. For the people who knowing the oral microbiology, and this is the place, and have been around for a long time, over 100 years, by a four Scottish brother, and who doesn't have their own children. So they decide to build a children dental hospital. We're the first children dental hospital in the world. And then they realize the same kids keep coming back, and drilling and the filling is not working. So they decide to become the MGH in dentistry. And they are the one decided to say, let's do the science, figure out the mechanism behind the oral disease. And that's how they invent the fluoride that have been, saved, billions of kids on suffering the tooth decays. And, but then the major oral disease are mostly periodontal disease, tooth decay, that are all bacterial infections. So that's why Forsyth suddenly become one of the, and the world leading center on, oral health, oral bacteria research institute. And because oral cavity is very easy to access, so we're ahead of a lot of like gut flora or skin flora, and in terms of understand, what's in those bacteria and what they're doing. And but something interesting happened is you probably all heard Harvard Medical School have a 15 affiliated hospital, and we are one of those. But there are only 14 now because last year, and I decided to make a strategic move. We become the first organization left Harvard and joined the American Dental Association. That was the title Boston Global describing this move, and called the biggest monster and in dentistry is created. And in many ways, this is very relevant because even though today I'm mostly focused on science, but in order to actually truly translating our science and into the technology, we realize we need a technology development, need a lot of money. And at the same time, the other thing is very important. It's the biggest barrier in dentistry. And for a lot of technology have not been effectively adopted is because ADA, American Dental Association, being the 800-pound gorilla in the room, they have not been truly supporting the innovation. Dentistry have been stayed the way it is. So recognize they are the true barrier. I just decided, we merge with them. Now we control all the clinical code, and we control the coda, and we're adding our innovation power to it. And so in many ways, I'm really saluting, Nikki is doing is because in many ways, publish a Nature Science paper, versus a position paper, white paper, and have creating an organization like this. and have an advocacy group is sometimes even more important and more impactful. And if we wanted to truly address the issue. And so this is kind of the things we are doing. And I'm very glad and here work with you and putting, breaking down the silos and bring the microbiome and Alzheimer's disease to have a true connection. So with this, let me focus on the science first, and then I will be talk about how we leverage the resource we have, especially our connection with the ADA and the supporting the effort you have. So every time when we talk about the oral microbiome, I always want to share this story. Do you know the very first microbiology paper ever published? And that was actually oral microbiome paper when Leeuwenhoek, took a tube and put it to glass on the side and tried to see it. He somehow smart enough and to think, oral must be very dirty. And he actually didn't clean teeth for 3 days. And this is the figure 2 of the first microbiology paper published. And as you can see from the very first observation of the oral microbiome, we already know the complexity of the system. And now 50 years later, we know all those bacteria. The little dots are the streptococci who responds mainly reason for tooth decay. And those long rods, the fusobacterium, is now become more and more famous. They not only causing a periodontal disease, and now they're one of the latest Nature paper you may see, the colon cancer, whether you get metastasis or not, is actually because this bacteria travel all the way to colon and directly determine the metastasis of that. Now, on this first observation, he also have is to find those sparkies. And we've been talking about the Lyme disease and the syphilis. And I'm going to tell you how much of actually those are in the oral cavity. It's a true jungle. And I'm very proud to say, and being the CEO of the Forsyth Institute, when you look at the history of the oral microbiome, where we evolving all the concept from studying the individual bacteria to taking the holistic approach. And we're the first one using Carl Wu's 16S concept and start to categorize all the bacteria in the cavity. So expanding, from we only know about 20 species to currently 700 species. From, taking the concept, Bill Costed and build it on biofilm, and then we're doing a lot of the ecology and ultimately leads to the oral microbiome. When you look at the evolution of the microbiology, and the oral microbiology is often taking the lead on that. And when you look at who are the leader behind this, or driver behind this, almost every one of the cases was have a Forsyth scientist in there. And that's why we're very proud. And even today you go to research.com and to listing the world's best microbiologists, and the limited few of the oral microbiologists listed on the top 1,000 microbiologists, they are all at Forsyth. So we made a lot of contribution to the field. I was, for Laura's sake, I will show you this because his father was built the first commercial anaerobic chamber. Forsyth is actually built the first manhood one because they were man-made one because they realized the oral bacteria is mostly anaerobic bacteria. And so back in the 50s, and this is literally a bombshell, and they converting that and into the first anaerobic chamber. Now, today, if you're reading this newest ones, NIH published about their 10 years of the work supporting the HMP, and the first paragraph, they are actually taking the oral microbiome data out of Forsyth as a milestone, go to the Congress to justify their $500 million spent on HMP, the Human Microbiome Project. And I'm also very proud is I'm part of the team and launched the Microbiome Initiative. I'm one of the 40 scientists at White House. And we were hoping at that time is to, this is again back to, saluting Nikki's effort is sometimes, we can publish papers, but we just realized efficacy is more important. And at that time, back in 2015, 16, so many of the funding agencies no longer funding microbiology. They think microbiology is a history. We went to the Obama administration to tell them the importance of the microbiome. And we made their Science and Technology Policy Office launched this microbiome initiative. Now it's become a household word. All the investors talking about it, or my wife is talking about it, and not from me, learn from me, but it's actually from the newspaper. And so I'm really hoping, you know, not for long, we're gonna be, we're gonna figure out a way to have everybody talking about how importance, you know, the microbiome and its connection with Alzheimer's disease. And I'm so glad to have such a diverse, you know, people and in this room, and I'm look forward to more interaction, how we can work together, really put this on the map. So one of the things I'm gonna do is I'm gonna give you some idea with all this 100 years of the cutting edge research we're doing on oral microbiology. We create a lot of resource, and some of the resource is only unique to Forsyth. And I'm here to say, we're open that things to everybody, and anybody have a good idea, wanted to, you know, moving deeper on studying the connection of the microbiome and versus Alzheimer's disease. So we really are here to help. So start with the diversity. And as I already mentioned, that there's 700 species, 20 trillion, 10 trillion, you know, and in the oral cavity. Forsyth have the best curated oral microbiome database and called HOMD, which is part of the support by the HMP. One of our greatest strengths is we understand in order to study the microbiology, use PCR to show its presence is not good enough. And you have to culture them. You'll be able to study them. You have to generate in the animal model to study them and all that. And we're so good at it. There are no any other microbiome, and they have a 70% of the bacteria are actually cultured and in our freezer. And that is very powerful, allow us to study anything when you use a probiotic and study their physiological function. And behind their success, we are the world's best place to culture the bacteria, anywhere the bacteria. And we have a lot of unique ways we're doing a nutritional mapping. We look at the genome. We have very good idea and what the bacteria nutritional requirements are. And if you can find a weird bacteria in the brain and you wanted to figure out what their brains are, how they get there, and we can actually help to isolate the bacteria, and we can study a lot of their work. Now, even with the best skill we have, there's still 30% of them are actually not be able culturable. And those are we call the dark matter and in the microbial world. And I wanted to share this one thing with you. Now, we haven't talked about this. That could be related to Alzheimer's disease. So if you look at the generally, you know, the eubacteria, the eukaryotes, the bacteria, and the archaea, and recently, and if you Google this word of CPR, Candida phyla radiation, and we actually had almost a fourth branch of the life, and they are made about 26% of the biomass in this world. Many of them are in the ocean, but you can find in the human body. And that entire phyla, 26% of the biomass, no one be able to culture any of them. And Forsyth is very proud that we be the first one. Actually, we cultured the first one and called TM7. And by this figure, you can see those little dots are the TM7s. And they're actually parasitic bacteria. And they will be small enough. They're only about 200 microns. So really between the bacteria and the virus. And it was a little bigger than the regular virus. And this bug have very small genome and less than 1,000 of the genes. And living in the interesting parasitic life. They were encoding actually 20% of genome with different suck system, so they can suck in the nutrition out of their food. But they're actually extracellular. And you can detect them in saliva, you can detect them in blood. They were directly related to the periodontal disease, and they were directly related to colitis and a lot of inflammation disease. That's where you can find one of the most abundant bacteria. Because they cannot be cultured and people have been ignoring them. And, but yesterday, the dinner conversation with Edward, and I learned the most common one you can find in TM7 is in dolphin. And apparently dolphin is the one known to have Alzheimer's disease. And that's the ones who run into the beach lost their direction. Just remember some of the mouse picture there. So, and this bacteria is very interesting. When they leave the main parasite bacteria, they become dorm. They're almost like a spore. They can survive on the bench without any nutrition for a very, very long time. And so that could be one of those bacteria related to Alzheimer's disease. I'm promoting it and hoping more people are actually looking into it. Or when next time you're doing the sequence, and if you find the TM7, let us know, because we know how to handle those bacteria. Now, the other things is amazing resource at Forsyth is I had this academy member, Gary Borrelia. He single-handed invented. He said, okay, sequencing is nice, but how deep you're sequencing, you lose the spatial information. And the higher sensitivity you have, the more contamination you have. You don't even know this is something from the contamination or real thing. Now, one of the real way you're gonna doing it is you want in situ, real time, and figure out what the samples are. And now we're doing the traditional fish. We only have to know what the bacteria is, and you design the right probe. But the Gary was figure out the way, because those modern imaging tool in combination with the different probe, which he called the classy fish. And the classy fish is let's see, there's only major 12 genera and in oral cavity. And he actually making a general probe against every one of the 12 genera. He will be able to light up the entire plaque and to figure out not only who interacting with who, and this has also become a discovery tool. And for you to understand in your section, your pathology section, you're wondering what those bacteria are, and using our spectral imaging system, and we could do that. And then the other things Forsyth have been doing is we finding bacteria, it's almost a microbiome, it's almost like a bacteria, a protein bag, inside of the cell. When we're taking 9 different sites, and even though you get 700 species, when you mix them up, you're gonna do it. But if you actually painstakingly picking different bacteria from different locations, you're finding at each site, the bacteria is very different. There is about 60 different streptococci in oral cavity. But when you go to the tongue, cheek, and the plaque, you're getting an entire different streptococci. And so that interests us very much. And we have this project that we call pan-metagenomic analysis. And you download every genome ever sequenced, line them up, and based on their, not only the genome, but also their spatial location. And the Forsyth have been finding, and almost every bacteria carry this area code zip code. And you, by looking at the sequence, you will know this is the bacteria is from the tongue, or from the cheek, or from the plaque. Now, I'm sure with all those other microbiome out there, if people manage to do the same thing, you're getting an ocean bacteria, you kind of know which feet they are. They're at the bottom of the ocean, or they're on the top of the ocean. So all those spatial information is super interesting. And we're even doing some of the genetic work to switch those areas and see whether we can actually switch and the tropism. And that actually might be able to allow us to identify whoever the one goes to the brain. And there may be, if we're gonna ever finding a brain tropic in our site, and those are the ones probably we'll figure out the mechanism how they go through the brain barrier. And certainly one of the biggest discovery at Forsyth, which I'm very glad the public can already hear us, is oral microbiome is a local infection by whole body dissemination. And when you brush your teeth and you see this microbleeding side, and you know some of the plaque is getting into your bloodstream. And given that those are such a strong biofilm, highly compact, they are moving around the bodies. And there are so many of the data have been showing preterm births, if it's not physically induced, 40% of those people, you can actually identify the oral bacteria in the amyloid fluid. And you probably heard from the news of the periodontal disease versus the heart disease connection was really for the same reason. Now, I'm glad that Cortexa is here because the Forsyth have been working with Cortexa and published that paper, 2019 paper, and on connection with the PG and detecting the gingipin, which is a Prophomonas gingivitis bacterial protein in the brain, and which is really indicating it's already evident to something what I wanted to talk about today. But given they're going everywhere, and there's so much of the evidence have been showing that is happening, and if they can go to the heart and the placenta, even though there's a brain barrier, there is a way to kind of go for it. And that was one of some of the things I want to talk about. I think the key question has always been, it's a direct effect or indirect effect. And here, all this great lecture today, I think we as a group have a very strong voice. It's a direct factor. And it's not because the bacteria is, induce information or anything, and maybe ultimately goes to the brain. So it's very interesting when you're talking to the people like Alzheimer Association Foundation, many of them still don't believe it. They always say, oh, brain barrier did a pretty good job. Bacteria never can pass it. And so my best answer to them is that you know nothing about infectious disease. Now, I teach 25 years at UCLA to a medical student about the infectious disease. And it's not a weather. And it's just yes, and the question we just need to figure out how, When I was an assistant professor at UCLA at that time, back in the early 90s, UCLA have, some of the best, Lyme disease and syphilis study, the scientists. And so to please the senior faculty, I've been working with Michael Lovett and Jim Miller quite extensively, on the Treponema pallidum and the Borrelia. And those are such a smart bacteria. They move around in the body, but they hide their flagella between the out membrane and the inner membrane because immune cell cannot detect it. And they can actually modify the immune system, not let them attacking them. They actually have I did the chemotaxis project, and they're running away from the immune cell. They literally know the hydrogen peroxide is coming. And when the tick bites, and no matter which part of the body tick bites, they always find the bloodstream because serum was the best attractant. And they always looking for the blood, where the blood vessel is, and they get into the brain. And I really believe that in a lot of cases, they have arthritis and others. We've been taking those fluid and from there, and you can always see they are the best attractant and for those bacteria and swimming there and accumulating there. Now, one thing we do know, and when you got those spirochete infection or some other meningitis, you have to use antibiotics to treat them early because anything late, they will eventually end up there. Any late stage of severe infection will causing a lot of problem. And when you look at all, those pathology data, we already know those bacteria are passing through the brain barrier. And so if those bacteria cannot do it, can do it, why not oral bacteria? And I'll just post those two questions here. Do you know how many oral spirochetes in your periodontal pocket? And how many Neisseria is in there? You'll be amazed by this number. Up to this point, 75 species of the spirochete has already been identified by the Forsyth scientists in our freezer. Now, every time when you have a tooth extraction, that dry socket, 95% of the bacteria in that dry socket is spirochete. They're seeking the blood, and they were swimming from every parts of the body to that dry socket and where you have it. And if they can swimming all the way to the to the socket, then as we always know from autonomy, and there are some major blood transporting pathway from oral cavity to brain, and they're so invasive, and it's no brainer for them to get there. Neisseria is another example, 10% of the total oral bacteria. They're making them a number 4 in the most abundant bacteria. Now, not every Neisseria is as invasive as meningitis, and but when they have the similar gene and genome and with the right mutation, with the right condition, and they probably will have the capability doing it. And certainly there are some specific evidence, and Forsyth is very proud and to be part of the team, published that paper and showing those evidence, and you can actually detecting a bacterial product in the brain. Now, there are many papers I can cite, and I just use this as example. And so the question is not about whether they can get into it. The question is really how they can get into it. The more molecular mechanism we have, and the more of the molecular detail and genetic nature we can understand, and the more that will make a difference. And I was just showing you one of the personal data I did. I'm a chemotaxis guy, and when you look at the spirochetes, the spirochetes have a very small genome as well, but they actually duplicate in their chemotaxis gene. And they have 2 sets of the chemotaxis gene. One is swimming in the environment, and the other one, they will actually swim inside of the body and to looking for which organ they're gonna be targeting to. So we made those mutant, obviously when then do that in our animal model. The non-motile one, when you get rid of the flagella and they can move around, they're not very pathogenic. That's not surprising. When we're knocking out their chemotaxis gene, even though they're actually still fully motile, they just do not know, and where the targeted cell is. In this case, they are actually looking for a dead cell, so they have the weak spot they can penetrate in the tissue. And you can see the Y type can actually zoom in the dead cell, but the mutant is all over the place. And when you injecting those into the mouse plates, and there's a much reduced rate and for them to penetrate into the brain, which is suggesting actually those bacteria are smart enough to actually recognize certain components. And if we're gonna be able to identify those chemoattractant, it will be a very powerful way and to treating the disease. Now one of the speakers, Dave's lab, have been talking about the polymicrobial infection, and which is a very popular topic in the periodontal disease. During the World War I, there's this disease they call trench disease. In 2 weeks, you lost the entire periodontal tissue, and they call ANAGA, acute necrosis, gingivitis. That have another name called fusosporosis. And we now actually know the mechanism. And the fusobacterium is can suppress the immune system, can induce the apoptosis of the cell. So they are very damaging, but they're not very invasive. And the spirochete is very invasive, but they're not very damaging. And those two actually figure out a way to work together. And when you have those very quick periodontal tissue destruction, which happens now in the AIDS patient, and in the old days, like I said, in the World War II, in the trench, where you have the poor nutrition and your immune system is down, the same thing happens. When those two gets together, the destruction of ability on the periodontal tissue is 1,000 times faster than the regular speed. And that is just give you the flavor of those synergistic pathogenesis, how important. There's some also very interesting social way. For example, we're talking about what happened to the PG and how the gingivitis will end up in the brain. That the PG with non-motile bacteria is actually all the way, and crawl there by themselves, or maybe hiding inside of the monocyte, get there, or there's some other way doing it. So one of our scientists are finding, and PG actually have a type 9 secretion system, which is bubbling all those extracellular vesicles. And gingiepin is one of the major components in that. And more interestingly, she finding all those bubbles have super sticky to the blood cells. And so they don't even have to get inside of the blood cell. They can stick on and use this as a vehicle and bring the extracellular membrane and to those other places. And I mentioned this Nature paper where we're part of the co-author and finding the fusobacteria, which is the oral bacteria, but directly responsible for metalysis in the colon. That mechanism is very well known now. It's they literally actually using the macrophage as a vehicle and allow them to get there. If they can get to the colon, that could be used the same mechanism, get to the brain. So I think, there's still have a lot of technology gap, and to make the rest of the community to, believe and agree what we're doing. And so I think, we probably all agree. And I think, Nikki will be good. We could do some consensus and pull out everyone to say what are the ones we all believe that's important. Then we can communicate in that with the funding agency as well, as NIH, and we should push them to creating a new RFA to addressing those critical questions. And but I do want to say Forsyth have a lot of those resources can help to address those questions. And I know my time is getting up, but I want to quickly cover that two other issues. And we have, I said, part of the reason we integrated with the ADA is we really finding, besides the science, we needed to do a lot of technology development and the health policy. And on the technology side, and we've been using a monoclonal antibodies, our sequencing system, and providing the saliva diagnostics. I was very excited to hear the bacteria you're working on can detect in the cats, and saliva. So we could be expanding our saliva diagnostics, into the pets as well, or beyond human. But one of the very exciting technology you may or may not heard of, so CRISPR, and especially the two CRISPR enzymes, CRISPR9 and the 13, and be able to amplify any DNA and RNA you desire without thermocycler. And so that essentially means right now, when you do PCR, you need a laboratory because no medical device can do it, at the disposable manner. When you use a monoclonal antibody, it's cheap, but its sensitivity is limited. But CRISPR technology is now changing that. You could actually getting a PCR sensitivity and at the price of the monoclonal antibody of cassette. And so we actually in licensed the technology, which is developed in the Broad Institute and Wyss Institute, and we're now providing them an oral application. And I think, as we're doing more of the oral microbiome connection with Alzheimer's disease, we could have those things ready to go. And it's just a simple design of another guide RNA, and the device is ready to go. So I'm very excited. This is an area we can potentially work together. We have been working a lot of other things, technology, and I'm finishing up. And I do want to say, we recognize money is also important. We're creating an ecosystem. Every year, Forsyth is doing what we call the DENTEC, which is investment forum. We bring all the people together, the investors and the inventor, and helping them to promoting it. And through the ADA, we can do a lot of education white paper standard and a clinical code and helping those technology to be successful. And I will quickly use one example. When we initially talking about the heart disease connecting with the periodontal disease, the AHA thinking, no, I'm not gonna allow a whole bunch of dentists coming into my territory. They publish a position paper saying this is all bullshit, you don't have the right evidence. And now, 12 years later, especially we're now part of the ADA, and this is a new initiative we jointly launched with the American Heart Association called Healthy Smile and the Healthy Heart. So I know, I hear all those news, you know, the major funding agency have not been funding it. And, you know, we may not be still all the voice, but hanging there, just like what we did with the heart disease. And we're actually getting there. And lastly, give me 1 second, is I'm play my CEO role, and we're moving to the new facility. Any great postdoc and graduate student interested in joining us, and it will be more than welcome to do so. We actually have an incubator and the VC funds, and we're actually supporting a lot of the technology, and the connection between Alzheimer's disease and the oral microbiome is one of our area. Even though we're very successful on NIH grants, and we have over 70 grants, and with over $100 million, NIH grant never can supporting the translational part of the activity. So we welcome a lot of the private foundation going to work with us to actively translating the science into technology. With this, thank you.
Thank you, Wenyuan. There'll be perhaps, as Nikki said, at the end of the session, time for questions. That was truly spectacular. So I'm now like to introduce Dr. Kevin Zwezdaryk and Sara Morris, a graduate student, who are gonna take over. So in issue of time, we're just gonna jump right in here.
Okay, everyone can hear me? Great. Hi, I'm Kevin, Sarah over there, and I am trained as a virologist, and there's a lot of neuroscience in here. And so my other team member is up there. Liz, can you wave, say hi? Half of these experiments would not have been able to be completed without assistance from her lab, so thank you. All right, so we're going to talk about CMV, a very common herpes virus, and how it may contribute to AD progression through metabolic pathways. So kind of the big questions in my lab are really, how do pathogens impact biological aging? And there's a number of sub-questions that arise from that, right? Is it the chronological age? And so someone talked about that earlier. Okay, is it the severity of infection? First time I got COVID, I got a phone call from work saying, hey, you're screening back positive. No clue I had it, right? Second time, I was in bed. Okay, so is the severity important? The type of infection. You know, you can break that down between viruses, bacteria, fungal infections. You know, is it a DNA virus versus an RNA virus, gram-negative, gram-positive bacteria, all of those things. Does that influence biological aging? A specific combination of pathogens. If you get hit with, you know, something that's a kind of commensal, so a herpes virus early on, and then a second pathogen hits you later or very soon after that infection, does it exacerbate the kind of long-term effects? And finally, what the focus of this talk is going to be is the number of times one's been infected over a lifespan. So is the combination of aging and viral burden important in this? So as I said, I started off as a virologist, really looking at how viruses change metabolism in the cells they're infecting, right? So viruses are obligate parasites. They don't really have a lot of genes that encode for metabolic pathways. So they're completely dependent on the host cells for all their metabolic processes. And at the time, there's some great work being done showing increases in glycolysis, increases in glutaminolysis, increases in fatty acid synthesis. What was missing and what I thought connected all those things was the mitochondria. All those pathways either feed into the mitochondria or out of the mitochondria. And so my lab was one of the first to do a real thorough characterization. There were some great papers that kind of led up to this by some other groups. But basically what you can see in the top is a seahorse assay that we saw earlier. And it's basically showing oxygen consumption rate. And so infected cells have a higher oxygen consumption rate, suggesting that the mitochondria is working harder during those first 48 hours of infection. The middle graph is showing mitochondrial membrane potential. And so if there's increased mitochondrial activity, the electron transport chain will be used a lot more. And so basically you're increasing the potential on that. And that's what you're seeing in the middle graph. And finally, if that electron transport chain is being driven, you're going to have oxidative stress in the form of reactive oxygen species, specifically mitochondrial specific superoxide. And that's kind of what we see at the bottom. So these cells are also experiencing oxidative stress. Kind of the big take-home message from all this is, remember, mitochondria have their own DNA. So what we did is we went and knocked down the DNA. So that DNA encodes for proteins involved in subunits of the electron transport chain complex. And so we had about a 90% knockdown of all those mitochondrial genes. And when you infect those cells, you can see that the virus has a very difficult time replicating, suggesting that the mitochondria is extremely important in the replication of these viruses. So I did all that, wrote my big R01. Study section was like, no, we don't care. Okay. And so luckily, I had a really good mentor, Michael Zwezdaryk. And that's what he told me. No one cares about this except you and the 4 other people in the world who are doing this. Okay. Make it more relevant. And he really said, look at where this data is leading. It's leading to pathology. Learn about some of these age-related diseases and what the underlying factors or associations are. And obviously, with the mitochondrial association, he said Alzheimer's is a great target for that. So we have a virus that's very common in the population. That seroprevalence increases with age. So it's 80 to 90% seroprevalence once you get over 70, 80 years of age. Almost 100% in third world or lower income countries. The reactivation frequency of herpes viruses have been reported to increase with age. So as you age, more and more frequent reactivation events. And kind of my work and a lot of other labs has shown that herpes viruses in particular, but all viruses, have these big metabolic changes. And so really, AD, a lot of people in this group and at this meeting have shown that AD can be influenced by changes in glycolysis, lipid synthesis, and oxidative stress. And that's kind of where my data was going. And so I changed my hypothesis and basically said, does CMV exacerbate AD through a metabolic pathway? The very first thing I had to do was create a mouse model. And my old graduate student, Mark Harrison, was instrumental in this. And basically what we did is took a very simple approach because common to this meeting, we didn't have a lot of money. And so we just used female BALB/c mice. And at 2 months of age, we would infect them with the sublethal dose of the murine version of CMV. And so after anywhere from 3 to 6, maybe 10 weeks, the virus is no longer replicating, but like all herpes viruses, it remains in the system and goes into latency. So a month later, maybe 6 weeks later, we can't really detect the virus at all. And we just kind of left those mice for 3 months. And then at that 3-month time point, we would re-challenge them with the exact same strain, exact same dose, exact same route. So we're kind of mimicking a reactivation event. Okay, and we kind of repeated this for 1 full year. So we went out to 14 months of age and we'd sack the mice. We also sacked them at the 1-month time point as well as a secondary control. So really trying to control for age, but also viral burden. And it was at this time that I had another student join the lab, and this is her work, so this is her story, and I will pass it over to Sara.
Can you guys hear me? Is that, oh, okay, yeah, we can. Okay. All right, so now let's get into the fun stuff. So this is single-cell RNA sequencing data that, oh my, I'm gonna hold this. Single-cell RNA sequencing data that from the brain. And as you can see, the cell populations are on the y-axis and metabolic genes are on the x-axis. And as I'm sure you can tell by all of the different sizes and all of the different colors, there are global transcriptional changes happening at the brain at the 12-month post-infection time point. Now, we know that MCMV does not cross the blood-brain barrier in adult mice. So moving forward, we decided to focus on the blood-brain barrier itself. Way too many things in my hand. And so moving forward, we're going to be focusing on CD31 positive endothelial cells from the brain. And to investigate glycolysis, we looked at GLUT1 and hexokinase 2. And as you can see at the 1-month and 12-month post-infection time points, there's no change in GLUT1 expression. However, when we look at VDAC1 as a proxy for oxidative phosphorylation, we see an increase at the 12-month post-infection time point. Now, this is going to be a reoccurring theme that you guys will see throughout this talk, that there's no change or minimal change at 1-month post-infection, but the magic really starts to happen at that 12-month post-infection time point. I'd also like to note that an increase in VDAC1 expression has previously been associated with Alzheimer's disease. So next, we used microvascular cells that we isolated from the brain and ran them on that seahorse assay that's been mentioned a few times so far. It's a live cell functional assay that uses oxygen consumption rate to measure various sorry. Anyway, at 1 month post-infection, again, no big changes are occurring, but at the 12-month post-infection time point, there's an increase in basal respiration, indicating that these mitochondria are having to work harder to meet the minimum requirements of the cell. There's also an increase in proton leak and non-mitochondrial respiration. This is indicative of mitochondrial and oxidative stress. So to validate these findings and look at them specifically, we did live cell flow cytometry to stain for mitochondrial mass and superoxide production, both of which are increased at the 12-month post-infection time point. And this is indicative of cellular stress. So at this point, another shout out to our collaborator, Dr. Angela Tarazi, and her lab, who have done a lot to help with the staining and cognitive testing we'll get into in a bit. And so first we stained for GFAP, which is the activation marker for astrocytes, and IBA1, the activation marker for microglia. And you can see, again, that 12-month infection time point, we're seeing significant increases. Next, we wanted to look at the permeability of the blood-brain barrier. And so we stained for claudin-5, a tight junction protein, and we also stained for the presence of fibrinogen and IgG in the parenchyma or brain matter. As you can see, at the 12-month post-infection time point, there's a decrease in claudin-5 expression and an increase in fibrinogen and IgG in the brain. Now, interestingly, this is happening in the hippocampus, but not in the striatum. Next, we did cognitive testing on all of the mice. And as you can see, using the Y maze, which tests for short-term working memory, the total alterations at 1 month post-infection are decreased, but they're making the same amount of correct alterations. Now, this flips at the 12-month post-infection, where they're making the similar number of decisions, but they're less correct. So far, we've shown that MCMV alters mitochondrial function in the cells of the blood-brain barrier. And this we showed with transcriptional expression of metabolic genes, increased protein expression of VDAC1, and increased mitochondrial function. And we also showed that MCMV impacts cognition. And again, I want to refer back to the model that we discussed previously. These changes are occurring with age, with repeat infection. And so now we're going to jazz up our model just a little bit. So my project primarily focuses on how MCMV infection will impact an AD model. We're using the 3xTG model. We're going to use 3 different viruses, MCMV as a systemic infection model, HSV1 as a neurotropic infection model, and influenza as an RNA virus, and a lung-centric infection model. We will keep the 2-month infection time point for the initial infection and reinfect every 3 months. The collection time points will be 1 week post-infection, 1, 2, 6, 10, 12, 16, 18, and 24 months post-infection. Each month, they'll be checked for weight, wellness score, and we will collect fecal samples for microbiome analysis. And at euthanasia, we'll collect an array of organs. What this will allow us to do is look at how the infection is impacting the brain, liver, immune system. We know that all of these things can be connected to Alzheimer's disease, and we believe that our model will help us understand the relationship and how they're interconnected. The next novel approach that we're using is an integrated multiome and spatial transcriptomics approach. And so at euthanasia, the brain will be collected and cut down the midline. The left hemisphere will be sagittally oriented on the z-axis, and the first 400 micrometer section will be used for multiome. Now, multiome is a RNA sequencing and ATAC sequencing. And then the next section of the brain will be used for H&E staining. And this will help us to normalize all of our results and ensure that every region we're interested in looking at will be available for spatial transcriptomics. So this is the result, our first results from the multiome. It's a heat map of differentially expressed genes. And as you can see, there are 4 distinct groups for our 4 different samples. So we have a mock male, a mock female, an infected male, and an infected female. And so as you can see, they are all very distinct, with the most similarities occurring with the females. This is really interesting to us, but not entirely surprising, as in the characterization of this model showed significant sex differences. And so seeing those present here is not entirely surprising. As you can see with transcriptional changes in both TAG and gene ontology pathways, there's significant changes, especially in neurodegeneration and Alzheimer's disease. And this is an H&E stain for the spatial transcriptomics that we'll be doing. I was really hoping to have that to be able to show you guys, but sadly, due to oddly enough construction, they are not ready yet. So hopefully you guys will be able to see that in a paper very soon. I would like to thank everybody who's worked on this project. It's really been a lot of people coming together. It's been a great thing to be a part of. I'd also like to thank our funders, IDSA, NIH, and the Tulane Brain Institute. Thank you so much.
Next speaker is Bill Eimer from Harvard and Mass General. He's going to be talking about his work with innate immune proteins in Alzheimer's disease.
Have a fine presentation.
Oh, yeah.
All right, that is very loud. There we go. For those of you who had the pleasure and the opportunity of meeting my mentor, Rob Moore, I just had to interject here and say, I think he would be blown away with the number of references to antimicrobial peptides. I met him in the last, it was at a neuroscience conference. He was the last speaker on the last day. It was an almost empty room. And as a brand new rising postdoc, I was like, this is amazing. And then we spent the next 9 years, as many of you have, fighting uphill to get things published and get funding. And this has been such a turnaround to sit here and watch the conversations around pathogens, antimicrobials, and this process forward that we're all trying to do. So I want to give a little bit of a background about how we got to where we got and where we've gone since then, looking at antimicrobials and Alzheimer's disease. So you have seen some of this before. Great minds think alike. The entire process over the last 30 years in its approach to Alzheimer's disease has been grounded in the amyloid cascade. This idea that we start with amyloid, it progresses to tangles, that progresses to neuroinflammation. That has been the driving factor in pharmaceutical choices, trying to find a way to either stop amyloid or stop tau in the hopes of stopping the disease, but that's sadly fallen a little short. So with this idea of pathogens, and I hope you are all convinced by this point that pathogens are least involved in Alzheimer's disease, we have to ask the question of what happens in between? Where does amyloid and tau fall in this path from contracting a pathogen to developing Alzheimer's disease? And I think I need to start with this fun little graph that Rob always loved. And it's looking at the phyla splits and different evolutionary splits between animals going all the way back 500 years. We're all the way at the top. And if we look right here, this lobe-finned fishes, coelacanth. This is considered a living fossil. If you're feeling very adventurous, you can go 450 feet down in the ocean off the coast of Southern Africa and still find this fish alive. It split from us about 350 million years ago. Its amyloid sequence is identical to ours. So you have to ask the question, if amyloid has no purpose, why has it been kept around for hundreds of millions of years? And our thought process was, well, Rob was sitting around with a Heineken or maybe 4 and looked at some related peptides, specifically LL37, and said, hey, it shares a ton of commonality with that. With antimicrobial peptides, there's a couple different conditions that most of them, not all of them, but about 95% of them share. They're between 12 and 15 amino acids, amyloid beta, check. They're charged, check. Takes on alpha helix, beta sheets, or a combination, check and check. So suddenly we're looking at this peptide that has no purpose other than to cause Alzheimer's disease, but it looks just like an antimicrobial and even aggregates just like an antimicrobial. So that's where it started. He took synthetic amyloid, applied it to E. faecalis, looking at a bacterium, and it managed to kill the bacteria. And so we went down with this initial model looking at how amyloid could act as an antimicrobial. We start with binding to the outside of a pathogen. It isolates that pathogen, protecting host cells, and then that leads to agglutination. And we tested multiple models and multiple pathogens in cells, in nematodes, in flies, in mice. We managed to find that when you overexpress amyloid and expose those model systems to a pathogen, be it Candida, be it Salmonella, be it HSV1, it was protective. More amyloid meant those models survived better. And in the one opportunity we had with Salmonella, when we took that amyloid away in a knockout, they had worse survival rates than our wild type. When we did our in vitro and really started looking at amyloid, just amyloid in a pathogen, we found that it was very effective and very quick at seeking out those pathogens, binding to them, and starting to agglutinate them. Even more so, when we looked very closely with Salmonella, the bacteria did not look healthy. So outside of anything else, just amyloid and bacteria, amyloid looks sufficient to start attacking and killing Salmonella. In some of my own personal work, we took HSV1, inspired by none other than Ruth Itzhaki and the 2 decades of work that she had done up to this point. We said, all right, it can do it in bacteria. Let's see if it can do it in an enveloped virus. And sure enough, within 15 minutes, we were seeing this tail of amyloid coming off the envelope of the virus. And by 30 minutes, it's already starting to form these agglutination of multiple viral particles. Even in HHV6A and B, we saw a similar pattern. Again, SEM that we followed, just like the bacterium, we see this clumping together and these little strings that you see here between the little circles, that's amyloid beta. When we took it into some of our living models, we have a 3D culture model that includes neurons, astrocytes, and oligodendrocytes. That model overexpressed amyloid, and by about 6 weeks, it would start forming plaques. It needs about 4 weeks to mature. We added HSV1 into that model right after it matured at 4 weeks. 24 hours, we had a plaque. And inside that plaque was HSV1. In 5XFAD mice, same thing. We wait till the mice are mature enough, they're reaching adulthood. That was about 2 months. If you keep watching, by 3.5, 4 months, you start seeing significant development of plaques. Within 21 days after infection at two months, we see plaques. And this is one of my favorite. We took the 3D matrix that we used to create our 3D cultures. No cells, just HSV1 and amyloid, and it was sufficient to create a plaque within that 3D matrix. Again, within 24 hours. So since that point, we have expanded our hypothesis and our model in terms of what amyloid is capable of as an antimicrobial peptide. We spent a lot of time showing this piece right here, that it can form plaques, it can agglutinate, it can protect by isolating. But antimicrobial peptides have a lot more abilities than just this. They can be involved in monitoring and regulating inflammation. They can be involved in apoptosis and managing the cell infections. So we started looking at the different pieces. Could amyloid target out infected cells? And in this simple culture, we had uninfected culture all by itself, and then we had a lightly infected culture where we could separate and identify infected and non-infected cells. amyloid preferentially found out those that were infected with HSV1, both in an overexpressing system and even in our naive system, we started to see a trend of that. What about when it's finding those cell survival? Is it targeting these cells and then helping manage the infection through killing off infected cells? And what we observed is, surprisingly, the first piece being that when we had amyloid and no infection, it actually had a protective effect. And then when we had just the infection, we lost cells as expected, but then compounded with infection and amyloid, the cytotoxicity of our cultures went up significantly. What about inflammation, a key part of Alzheimer's disease, and something that a lot of the antimicrobials can both upregulate and downregulate depending on the situation that they encounter? Well, we took our 3D cultures again, examining those that overexpressed amyloid beta and didn't, and with and without HSV1 infection. With just no infection, this group here examines upregulated genes as a result of amyloid. Then we have the effects of HSV1. We have the effects of HSV1 on amyloid, but then we have the effects of amyloid on HSV1. And interestingly, when you have both infection and amyloid, there is a whole slew of upregulated genes that don't occur without both pieces. And some of those targets are not surprising. TNF alpha signaling through NF kappa B, inflammatory response, apoptosis, STAT5 signaling. This starts to point towards the possibility that amyloid, in addition to isolating and attacking the pathogens, can also help regulate the immune response. And when we looked at the upstream immune canonical pathways, again, we start seeing even more pieces that play very well into Alzheimer's disease with inflammation, cell death, antiviral signaling, and senescence pathway. But Alzheimer's disease is not just A beta. Tau is also an essential part. And while A beta exists more or less outside the cell, tau is more or less inside the cell. But interestingly, tau loves to hang out, and its primary function is on microtubules, helping to stabilize them. And microtubules also happen to be the favorite highway for HSV1 to run up and down neurons. So we wanted to see if tau also played a role in this antimicrobial effect. And turns out it does. In a simple plaque assay where we cultured the cells, exposed them to GSK3 beta phosphorylated tau, the 2N4R, there's 6 isotypes, 2N4R and 3N4R are the 2 most common in the human brain. We found that that pre-incubation provided protection once HSV1 was exposed to the culture. We had fewer plaques, we had fewer individual infected cells, we saw fewer development from individual cells to plaques, and the plaques that did occur, the large ones, were actually smaller. But unlike amyloid beta, where we incubated the HSV1 with the amyloid before we applied to the culture, we had to pre-incubate the tau with the culture. Co-incubation did not work. We looked at the HSV1, once it enters into the cell, sheds its envelope, and that was the target of amyloid. So now we're dealing with the capsid. So we wanted to look at whether tau was interacting with the envelope or the capsid, and we found a significant preference for the capsid. And when we used an antibody against one of the structural proteins inside the capsid, we found a decrease in the tau binding, suggesting that it was sterically inhibited. Additionally, we tested the different combinations of tau in this binding assay and found that 2N4R GSK3 beta, the phosphorylated form, was the most effective at binding. Again, connecting back to Alzheimer's disease and hyperphosphorylation. In an EM, just like amyloid beta, when we incubated with the capsid, we started to see these tau fibrils connecting the capsid together. And inside our 3D culture, when we sliced it and took a look inside, these are capsids inside the nucleus of a cell, and the gold dots that you see are for tau associating with the surface of those capsids. We next kind of flipped the script. Instead of looking at how tau was interacting with HSV, we wanted to see how HSV was promoting tau inside these 3D cultures. And as expected, we saw increases in both dystrophic neurites and cell bodies that were positive for phospho-tau. The tau that was inside these cell bodies was actually leaning towards insoluble versus soluble. But what was very interesting was that while we were seeing an increase in the insoluble, the soluble actually seemed to be moving. The soluble p-tau seemed to be exiting the cell and entering into the media. So we thought that was kind of interesting, and we came up with this neat little experiment here to try to get a better perspective of what was happening. So we have a microfluidic device where we could enter the virus into one end and then allow it to flow across from left to right. And what this allowed us to do is control the expansion of the infection. And we could start to isolate certain sections of images. So in the red section here, these are infected cells. It's started to exit out through its retrograde axonal transportation right here at the edge of those microfluidics. The yellow area here is cells that are in close proximity but are not infected. And the green area is cells that are distanced from this infection and are also uninfected. And it provided an internal control for us. And what we saw was that the infected proximal cells had boosted p-tau levels, but the uninfected non-proximal cells were down at base level, and our highest p-tau levels were in the infected cells. But for total tau, it didn't change. The uninfected proximal and the uninfected non-proximal cells were the same. And when we correlated the signal of infected tau or infected cells around these proximal, it was highly correlated. The more infected cells you had close to these infected ones, the more p-tau that was present there. So to wrap up, we have expanded our model. We've now included tau in terms of this antimicrobial effect. And we're starting to see what I like to call my amyloid tetrahedron. Amyloid, tau, infection, neuroinflammation, they're all interconnected. They all play off of each other. Infection is boosting tau in response, boosting amyloid. Amyloid's trying to fight the infection. Amyloid's boosting neuroinflammation. Neuroinflammation's boosting amyloid. Amyloid's boosting tau. Tau's trying to fight the infection. It's all interplaying with each other. And there are many more pieces to this that we're trying to expand out and understand. And we hope that this kind of fills in that gap between a pathogen and the disease, and also provides a pretty strong reasoning for why pathogens are involved in Alzheimer's disease. So I'd like to thank both my mentors, Rob Moore and Rudy Tanzi, and the funding and Pulse PI for putting this together and making such great connections. Yep.
Sorry.
Just checking. Can people hear me at this level? A little louder? Okay. Just making sure. I don't want it to be too loud. Is this okay? Everybody good? Okay, excellent.
Well, that was incredible. Our next speaker is Dr. Laura Cox, also from Harvard University, and she's going to be talking about the role of Bacteriodes in Alzheimer's disease.
All right, thank you so much. So I really want to thank the organizers for the opportunity to share this work, but also to really sort of create a community where we can ask the tough questions, pull ideas together, and I think already we're making really great progress with the program today. So I think I have the only microbiome-based presentation. So I'll review some of the core concepts that are shaping our thinking. And then I'll show you some data which led to some surprises along the way. So the whole focus of my lab is to try to identify microbes that are altered in diseases that are really associated with clinical measurements of disease. Then to obtain those microbes and test their interactions in preclinical animal models with the hope of one day developing disease-specific therapeutics. And we do have a focus across a number of neurologic diseases, including AD, Parkinson's disease, dementia, multiple sclerosis, and ALS. We do think it's really important to understand these disease-specific interactions, and as well, potentially, even within the Alzheimer's space, understanding subtypes, different etiology and genetic risk. And so to think about the gut microbiome and how it could actually influence the brain while it's still present in the gut, it can really influence the brain in a number of different mechanisms. So I love this diagram here from Collins et al. And so the first thing is that microbes can secrete things like short-chain fatty acids that will induce our enteroendocrine cells to secrete neurotransmitters. But I think what's even more exciting is bacteria can make every single neurotransmitter that we know about. And so there is some debate, do these neurotransmitters actually make it into the brain? There's a study that shows a GABA-producing lactobacillus delivered by oral gavage can change behavior and make mice swim longer in a depression behavior test. There's also correlations with GABA-producing bacteria being depleted in the gut of subjects with major depressive disorder. So I think there is a translational aspect to the microbiota and some of the neuropsychiatric comorbidities. We know that microbes modulate immune cells in the gut, and those cells can then traffic to the brain. And this can have both a beneficial and a detrimental role. We're worried about too much inflammation in things like multiple sclerosis, reactive Th17 producing T cells. But other studies have actually found that if you infect mice with a worm, you can induce B regulatory cells that go to the CNS and put the fire out in models of MS. So there can be both beneficial and detrimental interactions with priming of the immune cells in the gut. We know the microbiota signal through the vagus nerve to affect pain responses and anxiety. And then it's also a two-way street. So if you've ever been like really nervous or anxious or excited and your stomach feels a little off, you're changing your gut motility with your neurotransmitters, and that can change your gut microbiota. And so turning to Alzheimer's disease, I think this has been an area that has only recently had characterization of the gut microbiota in AD. And this is one of the earlier large studies by Nicholas Vogt. And one of the things that we can see here is that there's an elevation in Bacteriodes, which actually correlates with A beta levels. So I'll talk a little bit about some of the concepts and frameworks and then go into some of the data from our own study. But we really think that the gut microbiota can be this thing that kind of mediates risk versus resilience with several hits along the way. We were having a nice conversation today at lunch about a number of hits. And in addition, we know that many factors shape our gut microbiota, including aging, diet, antibiotics, geographical location, lifestyle, and sex. All of those can change the gut microbiota, and those can influence both immunity and metabolism, which might ultimately influence Alzheimer's disease. So thinking about aging, there are studies that have directly shown using animal models, if you transfer an aged microbiota, it can lead to reduced neurogenesis, increased inflammation, but also immunosenescence, a reduced ability to fight infections. We see this double-edged sword in aging of increased inflammation and immunosenescence. This really does bring us into the concepts about how the gut microbiota could affect immunity in the brain. I think, unfortunately, the COVID pandemic has shown us that if you have a very acute inflammatory disease, that can lead to the development of dementia shortly thereafter. However, we also know that Alzheimer's is a very slow and chronic disease. And so I think that there is an alternate pathway in which a chronic colonization where you don't have this fulminant inflammation could lead to immunosenescence. And I think it's important for us to try to parse out those pathways and which pathogens are related to that. Animal models have shown that by modulating the gut microbiota with antibiotics can decrease plaques. And this was work by Sam Sisodia's group. And in about 5 or more papers from his group, they've consistently found antibiotics reduce plaques, but only in males. I think this next brings up the important factor of sex-specific differences. We know that women have a higher prevalence of Alzheimer's disease. They have a more aggressive disease course. And I think it's important to also understand that there's a really important relationship between our microbes and our hormones. Hormones are recirculated by enterohepatic circulation, and microbes that have beta-glucuronidase can cleave this beta-glucuronide tag. Instead of that hormone being excreted, you take the hormone back up into your bloodstream. And just sort of in common sense types of things, maybe things you've heard of before, it's anecdotally reported that if a woman is on oral contraceptives and goes on a high-dose antibiotics, the pill doesn't work anymore. So we know this to be the case, that there's this bidirectional communication there. And it's really hypothesized that sex-specific differences within neurologic diseases that emerge during different times in aging might be related to an aging microbiota. We also know that location is really important, and we can see that there's a different Alzheimer's prevalence by county, and this, of course, could be influenced by many, many factors, diet, pesticide, socioeconomic conditions, but I think it's also important to note that there is a differential exposure to both infections and antibiotics in the country, and that might also separately regulate Alzheimer's risk. And so 2 very clear examples, something that I know that's really important to this group. We know that Lyme disease, for example, is more prevalent in the Northeast, while the human plague, Yersinia pestis, is actually still causing infections in Arizona and New Mexico. And I think there was a recent case reported, I think just in the last month. So work that really started out of a fun conversation with my best friend during my PhD, who was studying metabolism and Alzheimer's disease. We wanted to ask, can we use dietary interventions to modulate the microbiota in aging and prevent AD pathogenesis? And so we started with the model of calorie restriction. We know that calorie restriction is linked to extended lifespan and protection from Alzheimer's disease. And so this study was led by Marissa Schaefer. And what she did, she introduced a 30% reduction in calories only from carbohydrates. We did not want to have malnutrition, so protein and fat calories were maintained exactly the same. And what she found was that it did have a dramatic protection on plaques, but only in female mice. So that was really interesting to us, once again, seeing sex-specific differences. We looked at changes in the microbiota over time, and what we found was that in our normal, the black line, the normal wild-type ad libitum-fed mice, Bacteriodes about doubled in aging, and in our Alzheimer's TG25-6 mice, it nearly tripled in aging, and it had the strongest correlation with A-beta plaques. There was also a loss in Faecalibaculum, and the calorie restriction diet normalized the levels of these microbes throughout aging. And so there are major concerns with malnutrition in aging, certainly with muscle loss, and it can have a lot of problems with comorbidities. And I think you also don't want to be hangry your whole life. So we're trying to find other strategies with the gut microbiota. And so again, it was a real surprise to me. I have a background in clinical microbiology. I know Bacteriodes can cause infections, but it's an extremely common member of our gut microbiota. So I had my doubts. So we looked into the literature. We found that Bacteriodes went up in aging humans, and that there seems to be a problem if you have too high levels in aging. So this study from Wilmansky et al. Found that in individuals who are 85 and older, if they had the third highest levels of Bacteriodes, they had a higher risk of mortality the next 4 years. And then again, the VOTE study found higher Bacteriodes. So to model this in mice, we delivered Bacteriodes to a faster model, this APP PS1 euchre mice, and we found that it indeed increased plaques in the brain. And this was showing that we were able to move from that correlation to causation in this model. The next thing that we wanted to do is find out how it's actually changing gene expression in the brain. So this is on a cortical tissue by nanostring. And what we found in our analysis was that it was modulating genes related to amyloid processing, but also GM-CSF signaling, which is a really important innate immune response, autophagy, neuroinflammation, senescence. Of course, we know that there's this classic amyloidogenic pathway generating plaques, and specifically, it was downregulating GGA1, which is a Golgi gene involved in clearance, and upregulating CDK5, which pushes this pro-amyloidogenic pathway. We next wanted to look at microglia, the major immune cell in the brain. And microglia in Alzheimer's disease can get this neurodegenerative disease phenotype, or this DAM phenotype, damage-associated microglia phenotype. And even though it's characterized or it's linked to Alzheimer's disease, these are both good and bad functions. You can imagine if you have a cut on your hand, you want all of those wound repair responses to kick in. And so we think that there's a beneficial role associated with plaque clearance, but also a detrimental role if they go into this unrestrained inflammation. And critically, we found that the microglia can affect, the microbiome can affect microglia across multiple neurologic diseases, but it seems to be disease specific. So most of the time, depleting the microbiota improves the disease and leads to less inflammatory microglia. In the ALS SOD model, antibiotics actually worsen disease and then actually increases microglial inflammatory phenotypes. So in this model, we sorted microglia from the brain, we sequenced it. And using IPA pathway analysis, we really surprisingly found that it was decreasing many genes related to phagocytosis, and it wasn't really activating a lot of inflammatory pathways. We looked at specific MG&D related genes, and we found that TREM2 and CTSL, CD33 were all being downregulated. And this was a real surprise to us. We had more plaques, but this MG&D gene signature was being decreased. And that was suggesting to us that Bacteriodes is suppressing the normal microglial repair processes. In order to test this more, we took aged wild-type mice. We treated them with Bacteriodes for 3 months. And then we did a short intervention of spiking in fluorescent A beta into the hippocampus and testing microglial uptake 16 hours later. And what we were able to confirm is that Bacteriodes directly inhibited this microglial clearance of A beta. And once again, when we sequenced those microglia, it was related to altering phagocytosis-related pathways, as well as GM-CSF, a cytokine that's really important for stimulating microglia. So GM-CSF is produced by many cells, but it is produced by T cells as well. We characterize peripheral immunity in splenocytes. We looked for inflammatory cytokines. We didn't see any. We didn't see any inflammation here. But what we did see is that GM-CSF goes up in our Alzheimer's mice, and then adding Bacteriodes suppress that level of GM-CSF. So once again, a story of immunosuppression. And so to summarize this work, we were surprised to find from our model that Bacteriodes is promoting A beta plaques by suppressing this immune-mediated amyloid clearance, again, suggesting that this chronic colonization could lead to immunosenescence, which I think was actually really nicely modeled with some of the chronic HSV work in the talk that we heard earlier today. And so just a few comments on Bacteriodes. It's ubiquitous and functionally diverse. We know that people the world over have a lot of Bacteriodes in their gut. And they're not all bad. There's a beautiful article from Cindy Sears, Bacteriodes, the good, the bad, and the nitty gritty. And we certainly know that there's important strain-specific differences that might mediate these effects. In neurologic diseases, Bacteriodes can make GABA. It has beneficial associations in depression. It has a polysaccharide A on its outer membrane. It can induce Tregs, and that might be good for MS. But there are some strains that carry this fragile lysine toxin, which is a matrix metalloprotease. It weakens tight junctions, and it actually interacts directly with PSEN1, an Alzheimer's risk factor. And so the next steps for my lab are to try to investigate strain-specific factors in Bacteriodes. And so to gather novel resources, we isolated Bacteriodes from healthy controls in Alzheimer's patients, grew up the bacteria, collected their supernatants and sterilized them, then conditioned macrophages with these bacterial secreted products. We then challenged with fluorescent A beta and then assessed uptake in this in vitro model. And what we were actually able to obtain is a number of different Bacteriodes strains with differential capacity to either stimulate uptake or fail to stimulate the uptake. And we're finding differential responses here. And so now we just finished this experiment. It was a 7-month experiment in which we colonized mice with AD-derived and a healthy control-derived Bacteriodes. And hopefully we'll have the brains cut and analyzed soon. So some closing thoughts on where do we go from here? Where does the field need to move towards? So hopefully we can find some microbiome-based therapeutic approaches. So just in general, we always could have subtractive therapy. Antibiotics have been miraculous, although sometimes they can be overused, some off-target effects. And I think the challenge there is to know what we're going after. Hopefully, if we can find specific agents, we could also get even more targeted with subtractive therapy using phage-based therapy or bacteriocins. There's modulatory therapy with prebiotics or microRNAs, which we find can selectively enhance specific microbes. And then, of course, there's additive therapeutics, including probiotics, FMT, or postbiotics, the microbial metabolites. And so for probiotics, you know, there's a limited number of microbes that are available commercially, but I think it is a rapidly expanding environment. The use of lactobacillus as probiotic therapy is over 100 years old now. And if it was going to cure Alzheimer's, we wouldn't have it. We would not have Alzheimer's today. So I think we need to be more targeted. There's mixed results on cognition, but again, I still think it's a promising area. And one thing to say is even though we think about probiotics being safe, in limited instances in immunocompromised populations, it can lead to serious bloodstream infection. So that's something to think about with a vulnerable elderly population. Antibiotics have been tried. A 3-month trial with rifaximin led to beneficial results. But again, there's problems with treating people long-term with antibiotics to cure Alzheimer's, certainly predisposing them to C. diff infections. And finally, there's been 2 case studies with fecal microbiota transfer where there was reversal of some of the cognitive decline. This was after having a C. diff infection. And again, there's some concerns with transmitting infectious agents, but I think this shows that microbiome-based therapies do have potential. And so closing slide, just thinking about our major goals and maybe things that the whole consortium and community of science can begin to address is I think we need to identify which microbes are contributing to Alzheimer's disease, and that will allow us to have these screening methods so that we can actually intervene before the cognitive symptoms occur. We need to understand how all of the different cofactors interact to help shape public policy and really have personalized medicine. We need to understand shared versus unique mechanisms across pathogens. I think, you know, we certainly could have those really hard hitters, pro-inflammatory microbes and more chronic immunosuppressive microbes. And so I think we need to understand how we design therapies within those subcases. And then finally, we hope to someday develop treatment guidelines for safe and effective microbiome-based therapies, especially within the Alzheimer's population that might be at increased risk of infection, even from probiotic agents. So with that, I'd like to acknowledge the many, many people involved in this work, and then also really say a big thank you to the organizers and IDSA. who gave me one of my first grants when I was trying to start my lab and really made a very big impact in my career. So thank you very much.
So our last speaker for this session is Dr. Monica Embers from Tulane University. Monica has for years been developing primate model of Lyme disease and Lyme borreliosis. So she's going to tell us about her work in this space.
Close to your face at first. Sorry.
Ow, my hair. It's okay. All right. Whew. Thank you for staying. I know this has been a long session. So I've been doing Lyme disease research for over 20 years now, and it's kind of, for me, it's kind of ironic that I'm here because neurodegenerative disease has affected my family. My father suffered from multiple sclerosis for many years and passed away in his late 60s. And now I'm watching my mother, my 80-year-old mother, go through Alzheimer's with sadness and curiosity. And so this has really been a good session. I want to thank you so much for inviting me. And I'm going to talk a little bit about, we've heard about Borrelia burgdorferi. So Lyme disease is a tick-transmitted infectious disease. It's the most common tick-borne disease in North America. And the great state of Pennsylvania is #1 in cases. So they're keeping it up. Symptoms oftentimes include a rash, arthritis, carditis, oftentimes neurological dysfunction. The number of cases in the United States, originally when I was writing my first grants, it was 20 to 30,000. And then a group at CDC decided to look at health insurance records and diagnostic test records. And then that 30,000 became 300,000. And right now we're well over 500,000 cases per year, just in the United States. So this is a big problem. And my lab has been studying Lyme disease, neuroborreliosis. About 15% of patients develop neurological disease. We've also focused on persistence, and we've shown in the rhesus macaque model that despite antibiotic treatment, the spirochetes can persist and they can invade the brain and they can remain there. And so this is very important to consider. Persistence has been shown in the central nervous system, the peripheral nervous system. And many years ago, way back in 1907, Fisher proposed that they were called senile plaques, looked like spirochetal colonies. So here you can see the plaques on the left and spirochetal colonies on the right. And as mentioned, Judith McCloskey did a lot of work looking at the connection between Alzheimer's and spirochetes. And she found very high percentages of patients or Alzheimer's brains that were positive for Borrelia. Just a number of cases here. Of course, the oral treponemes have been identified frequently. And I think what Judith came up with was a 25% of the brains in total through this review were positive for Borrelia, and there was a 13% or a 13-fold increase in the chance that you would develop dementia with a Borrelia infection. Now, I don't think that's fully accurate, but we've also seen Borrelia co-localizing with amyloid markers. Again, it's probably a useful antimicrobial peptide. And this was a really nice case study out of a group at Columbia showing Lyme disease associated with Alzheimer's. There have also been studies showing the opposite, that polymicrobial infections don't have Borrelia DNA, don't have Borrelia spirochetes. This particular report came out in 2017. There was no Borrelia detected in these patients. NIH study, again, looking in brain samples from AD patients, found no evidence of Borrelia. And then the arguments against involvement. And one important thing that I think that's been brought up is that you have to look at serology too, because there are a lot of these cases where they found Borrelia, but there was no serology to corroborate that the patient had Lyme disease. So there are confounding findings regarding spirochete involvement in the development of dementia. There's uncertainty with respect to the pathogenesis involving microbes. We talked about the role of beta amyloid. Also, what's the location of the pathogens with respect to the plaques and tau bundles? There's been a lot of new research looking at that. So I'm going to present to you two case studies that we have that are actually very different. So this is kind of our process. We get tissues from the Lyme disease biobank, and we section those, and we usually go 3 to 5 deep, where we do a big section, do PCR, and then do 10 micron sections for IFA and continue. And we use 2 different antibodies for Borrelia to make sure we're specific. We have a polyclonal and a monoclonal, and this just shows the merging of the two. 2 is always better than 1, but both of them are highly specific. We've shown that. So the first case I want to talk about is this Lewy body dementia case. So this was a 69-year-old woman who contracted Lyme disease at age 54. She had a well-documented EM rash. She was treated for 2 weeks with doxycycline, and she got better. She was positive on ELISA and Western blot, and 2 years later, started to become ill again. And it manifested as a sleep behavior disorder, and then she started to have cognitive problems, processing speed, mental tracking, deficits in visuospatial skills and executive functions. Once she had a brain PET CT, it showed decreased perfusion in the parietal and temporal lobes, and there were PCR tests for Bartonella henselae, Babesia microti, and Borrelia burgdorferi. They were all negative, but her Western blot was positive for Lyme disease. She was then subsequently treated again with intravenous ceftriaxone at age 80 for 8 weeks. And she improved to about 60%. And then she received oral amoxicillin, and that was continued for 6 months after the IV treatment. However, her condition worsened. The improvement wasn't sustained. She was given minocycline, and that there was no benefit to that. She had another MRI. It looked like she had some small vessel ischemia and demyelination. Finally, she developed severe oral dystonia and stopped eating, and she died 15 years after her initial diagnosis. And the clinical diagnosis was dementia with Lewy bodies. So Andrew Dwork at Columbia University did these stains, and on the left you'll see the histopathological findings showing primarily in the substantia nigra the Lewy bodies that were detected, and on the right the amyloid plaques in the patient's central nervous system. We were able to reliably and consistently amplify Borrelia DNA in the amygdala and the pons from this patient, shown with the asterisks here. And this is from the publication. This was a spirochete that was found in the spinal cord, actually, of the autopsy tissue. And it's very clean, very clearly a spirochete. And as we've continued to section this woman's brain and look, we find more spirochetes. So here's another spirochete that was found in her brain tissue. and another that's co-stained, which tells me clearly this is legitimately Borrelia. And the next case I want to talk to you about is a little bit different. It has a lot more to do with mental health. We just published this. This is a horrific tragedy that occurred. It was a young man who had multiple tick bites throughout his life. He turned to substance abuse because he became very socially, he had social anxiety. He was very depressed, and he started using PCP, phenylcyclidine, to subvert his symptoms. And there's a reason biochemically that worked for him. And he went through a period of withdrawal from that and went into a rage, basically walked next door to his best friend's house, stabbed his best friend, assaulted his best friend's, both of his best friend's parents, and then killed himself. And so we received his autopsy tissues, and we were basically looking for persistent Borrelia in this patient. We thought we would find it in the brain, but we didn't. We found clear staining of Borrelia spirochetes in his heart and in his pancreas. These are images, the top being the heart with our two antibodies, the monoclonal and the polyclonal, very clearly spirochetes. We also had a lab in Germany. We sent them the tissues, and they used fish, fluorescent in situ hybridization, to confirm what we had found. And we found quinolinic acid accumulation in the brain near the sites of inflammation. And so quinolinic acid builds up. It's part of the tryptophan kynurenine pathway, and as a result of chronic inflammation, patients can build up quinolinic acid, which is neurotoxic. And ironically, where this is an NMDA, PCP is an NMDA antagonist, and Borrelia can become an NMDA agonist. And so the fact that this kid started using PCP biochemically made sense because it was affecting his NMDA receptors. Oh, sure. So because there was a funding agency who said, Monica Embers is crazy, let's give her a project, another controversial project. You know, there's this different information about Lyme disease and Alzheimer's, so you need to study it, and we'll give you a little bit of money. So we did this study, and basically this is the analysis of AD brains versus non-AD brains for associated pathogens. We didn't just look for Borrelia burgdorferi, we also looked for Treponema denticola, C. albicans, HSV1, C. pneumoniae, and Bartonella species. And that if we had a positive PCR, we went ahead and sequenced that. And then in the sections where we did have a positive PCR, then we did immunofluorescent staining. And I'm going to jump to the conclusion here. These were primarily temporal pole samples from biobanks. And basically what we found was that there was Borrelia DNA in both the non-AD and the AD brains. If it has, if it has a pink highlight, that means it was sequenced. And we confirmed it. We found Bartonella in one of our AD. We found Candida in 2 controls, and we found Treponema denticola in 2 controls, and HSV1 in nothing. So it could just be the site. It's a needle in a haystack. But I think from what we found is that what I believe is that Borrelia can play a role in the development of dementia, and it depends on what area of the brain is affected, how the immune response, how the patient responds to the infection, which I think is very critical when it comes to Lyme disease. And because we've seen two different scenarios. We've seen the woman who developed Lewy body dementia, and we've seen the young man who, developed neuropsychiatric disease. And then the microbial etiology of neurodegenerative disease is supported by several lines of evidence. Obviously, we need further research. And we're continuing to evaluate autopsy specimens for Lyme disease and for Bartonella. And I can just say that there are several cases that we have where the patients had both Borrelia and Bartonella, and I think that's a very dangerous combination. And so my former graduate student, Remy Landry, and I produced this review article about dementia having a microbial cause, and we posited that there are 4 different characteristics that seem to pervade. Now, I know there are some exceptions because the blood-brain barrier permeability can be adjusted, but the first is the ability to cross the blood-brain barrier, tropism for CNS tissues and cells, the capacity to enter latency or a dormant phenotype, and of course, and I think very importantly, long-term evasion of host immunity. And so with that, I'd like to thank all the members of my lab, current members at the top, former members, Dr. Bransfield, Dr. Dwork and Dr. Fallon at Columbia University, and Dr. Liz Horn at the Lyme Disease Biobank, and of course, all the wonderful foundations who have supported us, as well as the NIH and the Department of Defense.
So I think we've got 5 minutes for questions. You can ask any of the speakers from this session. So I don't know if the speakers maybe want to come down here or something like that. And do we have any questions?
Also, we want to acknowledge that we're running a bit late. Some of the content was just so fantastic. I felt like we probably shouldn't pull people off stage, and people probably wouldn't mind going 20 or so minutes a bit later for sushi and wine and so forth. So we do appreciate everyone's patience with the schedule.
I actually have a question for our last speaker. So really, really interesting approach. And I was wondering, in the cases that were negative, where you're doing the PCR, are you also able to assess for known genetic risk factors? So you could kind of try to separate out like sporadic versus familial AD?
So we did have ApoE4 AD patients selected in that biobank in those samples already. So they were already predisposed. Yep.
And I had a question.
Yeah, please go ahead.
My name's Jen Thornton, and my father had Lewy body dementia, and we are 100% sure that a mite was the instigator. And mites can infest the nasal passage and swap DNA with humans, and can carry toxins, neurotoxins, fungi, Lyme, microbes, and dissolved tissues. Neurotoxins, as we know, can cause memory loss, dementia, and movement disorders. I was wondering if you are studying any mites connected with Lyme disease or mites with neurotoxins currently, or if anyone else is.
Did you say mites?
Mites, yes, mice can carry Lyme's disease.
Oh, mice.
Mites, M-I-T-E-S.
They
Like
don't.
dust
They
mites,
don't
like-
transmit
yeah.
Borrelia burgdorferi. Mites do not. They may transmit.
Yeah, there's a
Bartonella. I
research
don't-
study on it that they do. I have it documented in my book that I just published.
Okay, that's brand new to me.
Yeah, because we've had like people say, "Oh, I didn't get bit by a tick," but they did that have mite infestations and then end up with Lyme's.
Interesting.
Yeah.
What about some kind of a treatment protocol or prophylactic usage of medications? Out of curiosity, we do that with animals. Why don't we do it with us once in a while for some of these? I'm just curious.
Yeah, I don't know if this will answer that at all, but I was actually going to make a comment after Monica's talk. I gave a talk at the Medical University of Vienna a few years ago about, the increasing evidence from many of the people here, particularly with spirochetes and dementia. And this very elderly infectious disease gentleman got up afterwards and told me about 3 cases of Alzheimer's. They had been diagnosed with Alzheimer's disease that he treated, found out they had Lyme, and was actually able to reverse, their dementia, which I think is one of the goals actually of this whole group, the Alzheimer's Pathobiome Initiative, is to, identify, the constellations, the different constellations of pathogens, and then be able to treat. So I think you're speaking exactly, what the group as a whole, is working towards.
I guess along with those lines, Garth, is how do we specifically address that in a more rapid way within the timeline? Because with Lyme, I mean, my goodness, it's been 35 years for a test, as Monica knows. And there's a lot of other components here. I'd like to see something happen sooner than another 35 years. So that's kind of, I guess, some of the directive and question that I have.
So I think that's the major challenge that our field is facing today. I think one of the major things that it makes me more optimistic that we can actually design interventions is the new imaging, the new brain imaging, to actually detect amyloid and tau deposits decades before cognitive symptoms. And I think that's the time we need to intervene. So then if we're intervening, how do we do it? It's not going to be treating with antibiotics from the time you're 50 to hopefully 100. That's just not going to work. And so how can we think that there could be strategies either through specific metabolites, specific microbes to really finely tune the immune system. If we all think that infectious agents are contributing to AD, how do we keep them suppressed? How do we keep the viruses latent in the neurons and not coming out? How do we, prevent these microbes? Part of it might be exposure to ticks, and that might be something that is harder to avoid from a public health standpoint. And the treatment would be different for something with Borrelia, but with the endogenous viruses that live in us and microbes that live in us, I think it's strategies to promote effective immunity in aging.
So great question, if I may, is if you look at the whole pharmaceutical industry, no one developing antibiotics anymore. And so that is really the reason behind it, because the last one was Cubicin, and I don't know, 15, 20 years ago. And now I'm hoping through this new microbiome initiative, there are 300 to 400 microbiome companies. They will try to come up with a new solution, but most of the drug takes, 10, 15 years before really get on the market. So this big gap is a big issue. And, but I think, organization like this really crying for new technology is something super important.
Along those lines, and, our group, in addition, to studying perhaps, the infectious agents that are associated with this, have also been developing a new class of antibiotic, which doesn't kill. It actually shuts down virulence production. And so if you could shut down virulence production, that might be a completely different strategy you could use, because you wouldn't be killing, the good organisms. Okay, you're simply, as we say, defanging the pathogens.
So I was briefly mentioning, I built a company, we raised $250 million, and that was very much the concept. So bacteria actually, do their own sex. They have a lot of pheromones. We actually take one of those sex pheromone and put an antimicrobial peptide behind it. So I call the fatal attraction, and it was actually quite effectively doing a targeted killing and modulating the microbiome. So there are a lot of new technology from probiotics to phage therapy to slow nutrition to regulating it. So I'm still very hopeful. I think there's going to be a new line of antimicrobial will come out. And the other one was very exciting is the diagnostics, because if the diagnostics really picking up, that will be driving the therapeutics.
Well, thank you, everybody. We're going to take a 5-minute bio break, and then we'll come back for the last session. So, thank you.